is going to be fun, so it'd be a bummer to miss that, so. I don't know if we can squeeze it in anywhere else, so if we miss tomorrow's lab, we might have to just cancel it. Oh. I'll have to look at the syllabus, because pretty soon you'll be doing case studies. And then you're unknown. This semester is just rolling along. But fungi, we got to finish up thinking about fungi, unfortunately. I could go on and on and on, but I can't. So I think we left off around here with the idea that, you know, many fungi coexist with plants very intimately in the sense that they live their whole life inside the plant tissue and do not cause disease to that plant. We talked about Taxol, the drug, the billion-dollar drug that was discovered to also be produced by a fungal endophyte inside yew trees, in particular the bark of the tree. So that's a wonderful thing for the yew trees that it won't get cut down to extinction. And so also it's a definite boon to the company that makes this drug, because then you can just grab the fungus, get it growing in the lab, much easier to grow it and mass produce it much more quickly than if you're trying to get it from a plant and you have to wait for it to grow up enough to get enough isolated from the plant matter. So there are endophytes that produce the same chemical that the plant can make, like this idea, Taxol, both the plant and the endophyte. But there's also many endophytes that produce unique compounds that the plant do not. And so it's been very challenging to figure out what, you know, now that we know that there's endophytes, like who's responsible for this drug? Is it the plant or is it the residing endophytes inside? So that's a challenge, but it's exciting because we're discovering that there are endophytes in every plant that's been looked at, just like there's, you know, microbes inside of every human being and other animals. It's just this universal thing that seems to be, anytime you look, they're there. And so Dr. Strobel's work, when he was pioneering this endophyte discovery, he traveled the world and went and found all these really unique fungi. He did discover bacteria too, but seems like the vast majority of what he isolated and purified and studied were fungal endophytes. And you know, I think we mentioned in this previous slide that this endophyte, the Pestalotiopsis microspora fungus, this one is so easily transformable in the lab, which just means it's pretty easy to insert DNA into this fungus. And fungi can be tricky to get that to happen because they have a pretty tough cell wall. So it can be hard working with them in the lab. But so if, for example, you are trying to, you know, your business relies on the output of a typical secondary metabolite, like in the cannabis world, or all, you know, I can think about all the medicinal different types of plants that have been identified to date that are grown commercially. And then, you know, the secondary metabolite is what you desire. In many cases, it might be a smarter strategy to, if it's an endophyte that's really doing that, work with the endophyte instead of the plant. And so, especially if you can find an endophyte that's producing a secondary metabolite that maybe you initially thought the plant was doing, but actually it's the endophyte, or maybe they both make it. And if that endophyte is easily transformable, that could be a very big thing for the business owner, because then it's much easier to grow high quantities of the secondary metabolite on a much smaller footprint, much quicker over time. So it has a lot of benefits to thinking about, you know, their plants are famous for their ability to produce secondary metabolites, but now we know, oh, it's often the endophyte that's doing it, and they're much easier to work with. So if you have a little bit of microbiology experience, if you end up getting hired by somebody who's working with growing plants and using the plants, you may want to get them thinking about, well, are you sure it's the plant that's making the secondary metabolite? Maybe it's an endophyte, and we could capitalize on that and isolate the secondary metabolite from growing up the endophyte instead. So it's fun to think about from that regard. So knowing a little bit of microbiology can get you far. I think this is new material. So this is just the title of a published paper, and going back to the idea that endophytes have been there since day one. They now hypothesize it's just a part of the world of around us. I mean, that's the same with us. When you were born, you immediately were colonized by microbes. We'll talk more about that. Some even argue that when you were in the uterus, that you could have been colonized by low populations of beneficial microbes. And so that, I'll talk a little bit more about that. But anyways, 400 million years ago, they had a fossilized plant that they could see there was a fungal endophyte inside the plant fossilized tissue. So this idea that this is not something new. This has been going on. Microbes were here first, actually. When you think about it, they were here on the face of the earth first. And then all of us evolved in the presence of those microbes. So kind of makes sense when you look at it from that perspective. They were here first. And then I also wanted to point out, when you look at endophytes and think about their capabilities inside the plant tissue, they have the ability to move throughout the body of the plant. So they're often contained inside the seed. So the embryo, the baby plant inside that seed, often there's endophytes right there. And so they come with their own suite of endophytes. And then, of course, you plant the seed, it germinates and grows. And as that plant is growing, the endophyte can colonize different parts of the plant body. So it's not stuck in just one spot necessarily. It can migrate throughout the plant body. We also think endophytes, many endophytes, get into the plant through the soil. They're hanging out in the soil next to the roots, and they can get into the root tissue, and then they can migrate. So there's some really nice studies showing where they tag the microbe with like green fluorescent protein, and they can show the microbe traveling from the root up the stem into the leaves. And so you have this ability of the endophytes to mobilize and move in the plant. And then this idea that I was just talking about, the idea that endophytes might be a really good resource for finding commercial scale level secondary metabolites. And rather than trying to grow the plant, harvest the plant, and extract the secondary metabolite directly from the plant, switching into the endophyte idea. They're already doing this with yeast in a way where they're using yeast as a workhorse. Even in the cannabis world, they're looking at, can we get yeast, which are unicellular fungi, and grow them up in these huge vats and get them to produce THC. So they're looking at this idea with medicinal plants. So endophytes, I think you're going to hear a lot more about them. And it's really exciting when you start thinking about in terms of drug discovery and application of medicinal plants and their associated endophytes. One of my former grad students, Carrie Farkas-Lasich, who's now an assistant professor in our indoor egg program, she also was looking at mycorrhizal fungi and endophytes, but she really zoomed in on the endophytes inside this rare orchid. So this is, if any of you recognize it, if you by the Pictured Rocks area, there's big populations of this ram's head lady slipper, very beautiful orchid. And Dr. Robertus's students discovered that in that area of Pictured Rocks, there was a couple million of these plants, individuals, where it's going extinct or has gone extinct elsewhere. So we have a huge population here in the Upper Peninsula, which is really weird. And it's in sand dunes. So it's like, what's so great about that they're thriving in the sand dunes of the UP, but going extinct in other parts of the world. And so Dr. Robertus's students were able to discern that information, that there's all kinds of individuals. And then Carrie was helping out one of her name was Adrienne, which was Dr. Robertus's student. And so when the story started to play out, we're like, well, what's going on? And of course, from my perspective, it's like, I think there's something going on with either endophytes or mycorrhizal fungi, below ground activities, perhaps in the roots, where for some reason in that habitat is allowing these ram's heads, lady slippers to survive. And so really interesting studies that were done on this particular plant. And the story is still unfolding, but Carrie did find some unique endophytes that were popping up and, she sampled. And by the way, she was allowed to sample the tissue because there was so many of the individuals here, it wasn't at risk of, making the plant risk going extinct. So that's why she was allowed to she had to get special permission and get a permit. She did everything. And the reason we were able to do that is because they documented there was so many out there. It wasn't a risk to take some samples and take them to the lab. So she did a really nice study looking at endophytes. And we think some of these endophytes were very helpful in the ability of these ram's head to survive a very sandy environment and windswept environment. So lots more study can be done on that whole topic. Lots of interesting questions yet to ask. There's one for you. Can you read that? Without farming, you would be hungry, naked, and sober. I didn't take that picture, but I thought that was very appropriate. So, of course, a lot of my research is looking at how do we feed so many people on this planet and do it in a way that when our soil systems are pretty much maxed out, we don't have great soil anymore available. We have marginal soil. That's why I like the idea of indoor agriculture, take it inside. There's lots of vacant buildings in cities that can be transformed into this idea. And then you get locally grown food, very quickly grown food compared to this. And you don't have to worry about the climate issues in terms of we have winter for so long here. It's really short growing season. If any of you tried to stay up here over the summer and grow a garden, it's tricky. I still can't get peppers to grow. I mean, I'm trying to do green peppers or nope, I can't get them to full harvest by the time it gets too cold. But anyways, fungal endophytes, mycorrhizal fungi, all of that is playing into the future of farming. This is a book, just the title, the surface of the book, Microbiome Stimulants for Crops. So they're looking at that idea of we have to get the plant to have a nice plant microbiome, lots of good combinations of fungi and bacteria. And they're really, really zooming in on this now because so many of their they used to rely so heavily on chemicals to protect the plant and get the plant to harvest. And now a lot of those chemicals are being phased out because they're too toxic. They've done too much damage to the environment and to people. And so now they are scrambling. What else can we use to help plants? And so fungi, mycorrhizal fungi, endophytes, this has become a hot topic because we kind of force the situation that they have to look at things besides, pesticides and herbicides and things like that. Certainly in the food industry too, they're looking at growing fungi and having that as a major source of nutrition for humans. So this picture here is Nature's Find company. Is a company, I don't know where they're based actually, maybe in California, but they discovered Fusarium flavolapis, flavolapis fungus out in Yellowstone. Yellowstone? Somewhere. No, no, not Yellowstone. Where was it? The one with all the hot, it says it on one of the next. Is it Yellowstone? That's the one with all the hot springs and yeah, okay, that's what I'm thinking of. Yeah, from the geysers of the West to the food capital of the it's Midwest. Grown in Chicago. So that's where their company is. Anyways, they isolated this fungus from that national park, started studying it and realized that it's got a complete protein source in it. Good source of vitamins, minerals, and fiber. So I did a dive online to try to find out more about this company, and they're keeping everything very close to the vest. They don't want to explain how they grow this fungus. It's their baby. They want to make this their business. So, but that's it growing in like a kind of a pancake-like growth form. And my guess is what they're doing is they have like like those roux bottles I talked about. They probably have a film of liquid that they inoculate the fungus, and it grows like a carpet across the liquid media. And then they can take that and you can read more about this on your own if you're interested in. But they can take that and, feed it the right ingredients to give it so that it has a complete set of protein by the time it's ready to harvest, and the fiber and minerals, no cholesterol or trans fat, vegan. And so then what they do, and then they say, and then when we grow it, we're using hardly any land because it's all indoors, and it's, you can go vertical with it. And of course, you don't use the same kind of water you do when you grow crops, but they turn it into different things. So they're turning this into cream cheese. They're turning it into meatless sausage patties, like breakfast patties. So has anyone seen Nature's Find on the store shelves? I have not. So I don't know where it is I know that they have done limited sales here and there, like pop-up, like, hey, we have this for sale right now, and then they get sold out. And so I think they're early in the stage yet on getting it mass produced, but it sounds like this could be very successful. So you're thinking about things like this where you can grow vertical, and they're looking at not just doing it with this fungi. There are others that are in the pipeline too. Yep.
I was just wondering that like actually it's just like kind of keep the mycelium.
Yeah, so they take the mycelium and work with that. And then, you have to, I'm sure they employ a lot of food scientists that, okay, how do you transform that into the cream cheese versus the sausage? That's very different. And yet, hopefully they're maintaining all the quality of the nutrition, nothing's getting lost in that. Different ways of transforming it. But even, Hollywood actors are getting into this where they're looking at funding some of this. And so some of the companies are in that stage where they're just trying to get the funding going and see if they can raise enough money to start out. My Bacon, that's the name of it, My Bacon. Has 6 ingredients: mycelium, coconut oil, cane sugar, sea salt, smoked flavors, and beet juice. And it's supposed to taste like bacon. What do you think? Did you try it? I think it'd be worth trying. I mean, that could be huge, way healthier than bacon. Real bacon. So, and then locally, Myconaut. I don't know if you've heard about that company. Joe Lane and his partner Ryan Iacobacci. And Joe did his bachelor's here at NMU. I was his advisor. He was my advisee. And we've had many, many conversations about fungi and how to grow edible fungi. But so Joe and Ryan got together for this Myconaut business that they have. And they are using the Smart Zone Invent NMU process to get their business up and running. And they've submitted grants to the National Science Foundation and other granting entities. But basically, they're looking at fungi for edible food, medicinal, and now they're looking at PFAS ideas, how to clean up the PFAS issue that we have everywhere, apparently. And they're also teaching in the indoor egg program. They're helping out with the commercialization of how to grow, fungi commercially. So they gave a presentation. They won the what was what was it called? The Pitch Prize. Where was this? Can't remember. I think this was down in Chicago. They had a competition and they won the Pitch Prize down there, and that was on the PFAS idea. So, there's other companies besides Myconaut, but this is a local one with some of the, Joe being a former biology student. So, I think now more than ever, this idea of the use of fungi to help with remediating contaminants as well as medicinal and edible, it's really becoming a thing, which I just love. I love that idea. I'm excited. It's about time they got their time in the limelight, sunlight. Look at that fungus. Beautiful. So this one is Lactarius indigo. You can see why they named it indigo. But it really can have that deep of a pigment. And when it, I saw another picture where it was kind of oozing liquid and it was just blue. Like a, kind of like a latex like pigment, but instead of white, it's blue. So this is an ectomycorrhizal fungus. It's edible, believe it or not, that is edible. And of course, I don't know, if I saw that at a farmer's market or something, wouldn't you be attracted to that? Like, what is that? I mean, it's so eye-catching for humans. So it's a popular food, edible food. And this paper I thought was great. A novel approach to combine food production, carbon sequestration, biodiversity, and conservation goals. So their pitch is, why don't we, instead of cutting down the forests, especially when you think about how some forests are cut down so cattle can end up grazing on that stretch of area. And usually it's only good for 1 or 2 seasons, and then the soil can no longer support the grasses the cattle feed on. So it's just ridiculous. So what if you keep the forests, don't chop them all down, you stimulate that system to have this fungus in it, which isn't too hard if you get the right trees and plants in there. And then if you inoculate the forest soil with, spores of this ectomycorrhizal fungus, you can get colonization. And so they did this study and they were able to then equate it to, how much protein would you get from growing fungi instead of grazing cattle on a deforested area. And so, all in one big idea, they're getting at all of these ideas of fungi can sequester carbon below ground, so that's great. You keep your biodiversity because you haven't chopped down the forest. And your conservation goals, of course. And so that, I'm not asking you to read this paper, but I just thought when I saw that published, when was that? 2022, so not that long ago, but this is what they're saying, you could have your forested site or, afforestation just means that you're kind of creating a forested site. And then it gets your inoculum, your fungal inoculum, and start getting the plants, the trees and the fungus together. And then when the fruiting body comes up, you harvest that. And so they're saying you can get, here. It may enhance biodiversity, contribute to conservational goals, create a net sink of greenhouse gases, while at the same time producing a similar or higher level of protein per unit area than, well, if you compare that to grazing cattle per kilogram, you can actually get very similar results. So these are the types of ideas I think that are really going to help solve some of our problems that we have currently and into the future about the loss of forests due to cattle production and other animal systems that are being used. So lots of creative thinking going on there. I think it's great. So, okay. Any questions on any of the fungi stuff? Well, I encourage you to play around with fungi. You can buy fungal growing kits that you can put right on your kitchen counter. And then all you basically have to do is spray them with a spritzer of water, from a water bottle every now and then, and watch them grow. And then you can eat them. And super healthy. You can get creative with different ways to incorporate them if you don't like the texture. There's ways you can hide that if you don't like the texture. So lots of different flavors depending on the species. So it's a lot of fun. Maybe ask that for Christmas presents from your parents or something. So right now in Plant Kingdom, that's the other class I'm teaching this semester. We're growing edible fungi right now in the lab. So 6 different species, including shiitake and lion's mane. So lots of fun. It's fun to watch them grow. And I put it right on the kitchen table at my house, trying to get my boys brainwashed. Like, yes, fungi are awesome. You should eat fungi. They haven't quite got there, but they're getting there. Just got to disguise the texture. So if anyone has a good idea for that, other than soups, like pureeing it,
blending it up. I know that could be one method, but I think fry it. Yeah, pretty much anything's good fried, right? Isn't that the joke at the Minnesota State Fair? Everything's fried and on a stick. I think that's even butter. Don't they do that there? Fried butter? Where they have the coating on it. Haven't tried that. All right. Fungi are awesome, but now I got to shift over into the next topic. Which is viruses. Which, believe it or not, viruses, there are many, many beneficial viruses. I know that's gonna not sit well with so many people because all we hear about are the nasty viruses. Granted, they're impressive. I can understand why they're so newsworthy, but the vast majority of viruses out there, I mean, right now, there's viruses in this air that aren't pathogenic to humans, but we're exposed to them and they're no danger to us. The calculations of how many viruses, there's a New York Times blurb. The title is something like trillions of viruses fall from the sky every day. Kind of a shocking title, but it's true. They've documented it. And the vast majority of those viruses want nothing to do with us, so to speak. So in this topic, I certainly will talk about some of the bad ones, some of the pathogenic ones. We need to think a little bit about their structure and function, but I also want to introduce their ecology in the sense of they don't just cause misery for humans and other animals. There is plenty of good things that they're doing in an ecological context that's important for how our planet works and in the diversity we see on this planet. All right, but let's get down to some of the basic ideas. So this is just showing some images of either the virus itself or you can visualize viruses on auger plates. So they kind of they don't form colonies. They call them plaque forming units. So we'll talk about that, like how do you do that? Actually you have them on an auger plate. And then you can see that there's some nice images here under the microscope. So you have to use an electron microscope in order to see them. Unless you're just seeing the presence of them, like on this poor baby or on the leaf. So symptoms you can see, but if you want to see the actual virus, you have to get an electron microscope. So viruses are not, most people would not consider them living entities. So we tend to talk about viruses like we'll say virus particles. Because if you look at how they're constructed, they don't have all the qualities that we define as something that's living. So if you look up the definition of what is a living organism, viruses don't fit all the criteria. We know that viruses can exist outside the host, but they can't reproduce themselves. We've yet to discover a virus that can replicate itself outside of the host, whatever host it might be. We often will talk about the ones that are outside the host, especially virologists like to use the word virion. I really don't use that word. I'll say virus particle whenever I'm referring to just, the actual virus that we're staring at. When they're outside of the host, they're not active in any way. They are just kind of sitting there waiting for hopefully their host to come by. So the fancy way to say that is they're metabolically inert. And viruses that infect us have a challenging job because not only are we multicellular and complex with many different organs and body parts, so the virus has to get to the right tissue type that it can actually infect, which is challenging. We'll talk a little bit about that. And then not only do they have to get to the right tissue type, then they have to get into the actual cells, which is challenging. especially when we have such an amazing immune system. So basically, they take the cell, the host cell hostage, and they basically say, just make more of me. That's all I want you to do. Make more viral particles. And then they usually bust out and break open the cell and the cell dies. Not always, but usually. And so they'll use the enzymes of the hosts mostly. Sometimes they bring their own enzymes. But most of the time, we see them take advantage of the enzymes already up and running. They'll take advantage of all of the necessary machinery to do transcription and translation. So they really take advantage of what that host has to provide in terms of not only physical space, but all of the machinery.
Say, they bring their own enzyme. The enzyme or the enzyme? It's
Both, it depends. Sometimes the virus has the enzyme inside it, we call it a capsid, and sometimes they have the genes. Yeah. It just depends on the virus. Viruses are amazingly diverse, and the more they study viruses, the more surprising they are. This is just a mock-up trying to show, you have to think really small when you get into thinking about viruses. This is like only a partial drawing of a eukaryotic cell on top, and then you have a prokaryotic cell over here to the left. And then the T-even bacteriophage, which looks like something out of a bad sci-fi movie. I've actually seen a sci-fi movie where they have that as one of the evil things that came to Earth and was taking over Earth. I can't remember the name of it. I was in grad school. It was like, one of those B movies that never made it. So I figured, hey, see, if you're a microbiologist, you can get a job in Hollywood, because they had to have taught to a microbiologist or a virologist to get those types of ideas. So anyways, and then the picture next to it is an electron microscope picture. They did color code it so that you can make out, the difference of what's going on with the virus particle. And here, what's happening is it's attached to a cell. And I don't know if you can see from the back of the room, but that squiggle there, that's the DNA that it is injecting into the cell, very much like a hypodermic needle or syringe. So viruses, different morphologies and shapes. Some are very simple, some are very complex, like that one that looks like something from a bad sci-fi movie. And so before the HIV, before that virus, the tobacco mosaic virus is probably the most heavily studied virus, because it's easy to work with in the lab. You don't need any special permits or safety issues, because it only infects tobacco plants or related plants to that. So it's okay to work with. It doesn't infect humans. So they just trying to understand viruses. Tobacco mosaic was probably the most widely studied until HIV came along. Here's some other images. So COVID is the lower left, that virus. And here they have a nice depiction of that T4, T2 phage. And so you can see they can be quite complex, where they actually have different body parts, so to speak. And look pretty fancy. So when we think about all the viruses, some of the common properties is that they tend to have some kind of protein coat, whether it's very simple, like a tobacco mosaic virus, it just looks like this long rod-shaped thing, or that very fancy bacteriophage we were just looking at. The capsid is basically a bunch of subunits of protein, and it is so it's kind of like their version of, they don't have a cell membrane or a cell wall or anything like that. So, but they have to have some kind of barrier to the outside world. And so the protein capsid is that. The slang for that is coat, protein coat. And then if you look inside that, if you crack that open and look inside, what you would see is some kind of nucleic acid. So they say in the core of the capsid, you'll find the nucleic acid. And depending on the virus, it's either the nucleic acid is RNA or it's DNA, but it can't be both. There's yet to be a virus discovered that carries both RNA and DNA. I just haven't seen that. What's even funkier is that the nucleic acid can either be single or double stranded. So, when you took BI 111 and all these other classes you're taking, we always tell you, DNA is double stranded, messenger RNA is single stranded. Well, viruses have to be different. They can have RNA that's single stranded, but they can have RNA that's double stranded. DNA too, they can have double stranded DNA or they can have single stranded DNA. So they break the mold when it comes to the nucleic acid paradigm that we always teach about in basic biology classes. And then depending on the virus, not every single virus, but some, they can have some of their own enzymes that they'll bring with, or genes that code for that. They can have their own version of a lysozyme enzyme. Neuraminidase, which is a very hard word to say, helps them break down tissue as they're doing their infecting of the host, especially an animal. RNA polymerase, they can have enzymes that will serve as RNA polymerase, which is helping to make the messenger RNA. They can bring reverse transcriptase to the game. So that's a very special enzyme that can take the information on RNA and make a DNA off of that. So very, very funky. Viruses that are successful attacking animals, including us, very often they have this additional thing surrounding their capsid that we just call a lipid bilayer. The virologists like to use the word envelope, but really all it is the lipid bilayer is actually taken from the host. So as this virus is exiting out of the host cell, it can grab the membrane, part of the membrane, and kind of wrap that around itself. And then they'll stick their own proteins in that as to kind of like make it their own. They accessorize, so to speak. Can you think why a virus might have evolved that capability? So as they're sneaking out of the cell, they wrap themselves in that cell membrane. We have a very sophisticated immune system. So I like to think of it as the Harry Potter and, the cloak of invisibility that Harry Potter had. That's kind of like what I imagine the virus laughing as it wraps it around, wraps that membrane around itself, because then your immune system has a much harder time discerning that there's anything wrong, because it looks just like the human cell membrane, maybe with a little bit of variation. There's usually some specific proteins coming from the virus, but very, very nice adaptation for those that attack, especially animal viruses that attack animals. So based on some of what's listed here, that's how virologists will try to figure out how to classify viruses. And like, they don't have, domain, phylum, class, family, order, gene. They don't do that. Instead, they have this Baltimore virus classification system, and they have them in different groups. You don't need to memorize this, but you do need to think about, well, how are they classifying them? And it's basically one of the first things they look at is what is their nucleic acid. And so you can see right here that they group them depending on, are they double-stranded DNA or single-stranded? Are they double or single-stranded RNA? So you don't need to memorize which group is which, but know how they're doing that. It's by the nucleic acid type. And then certainly they look at the overall kind of design and morphology of the virus particle as they're trying to figure out and learn more about it. So this is trying to get across the point how the capsid is made, that it's a bunch of subunits of protein that gets all put together to create that capsid design. But and then if you look inside, you'll have that the nucleic acid inside of it. Here's another mock-up just trying to show, and they actually use these words, naked. Naked virus means that it doesn't have any envelope around it. An envelope virus means that it did steal some of the host cell membrane, or it might be an internal membrane that they take as well. It doesn't always have to be the cell membrane. But again, you're looking at the anatomy of the virus particles, and that they can have quite a bit of difference to their designs, their overall design. But they all have some kind of nucleic acid that's encased in some kind of capsid protein material. So sometimes very fancy, many-sided. Sometimes like the tobacco, it's just pretty straightforward. So when you can look at these under the microscope, sometimes they're quite intricate. You might have heard of these. These have been hitting the headlines over the recent years here with climate change and how the tundra is warming up. The frost line or the freezing line is not so deep anymore, and it's releasing viruses from the permafrost area. And which has a lot of virologists concerned about what does that mean? How, if we get exposed to some of those viruses that we haven't seen for millennia, is that going to be a problem? And so there's a lot of people, I'm sure there's probably a movie along with that plot line somewhere out there or in the works of, another issue that we have to think about. So giant viruses are often found in those types of areas in the tundra. They've discovered them. This giant virus apparently is not from the permafrost. It's affecting an amoeba, apparently. Mimi virus, you might have heard of Mimi, M-I-M-I virus, Mimi virus, that one. Where is it? It's as large as some bacteria. That's pretty amazing. And then the Mimi virus is actually can get infected by other viruses. So you have viruses infecting viruses. So it's like a nesting doll where you have the virus infected with the virus, which then can infect the amoeba. And it's just as big as a bacteria. So we get into some wacky things when you start looking at viruses and what they can do. It's who needs science fiction if you're a biologist or a microbiologist in particular? You don't need to read science fiction. It's all here in some way, one way or another. So they're studying some of these giant viruses, and this is from the Antarctic regions when they're looking at the permafrost areas. The Siberian tundra is melting. So that lower right image is called a pithovirus. This one is weird because it actually seems to carry some ribosomes. That's the first they've ever discovered. They've not seen that in any other virus particle. And this has been, frozen in a tundra for a long time. So there's probably others out there like this. They just haven't discovered that in anything else yet. So I'm sure there's a lot to discover yet when you're looking at viruses, especially when you start going out into different ecosystems and hunt for them like this, and not just focus on the ones that cause misery to humans. So definitely giant viruses are becoming a thing. It wasn't just a one-off when they first discovered the very first one. They're now finding, oh, yeah, there's actually more out there. And I'm not going to get into details on this, but basically we're looking at this idea that it really starts getting you to think about the evolutionary process and like, do viruses show up first and then cellular organisms or vice versa? How do viruses impact the evolutionary process? Here they're actually saying maybe viruses actually came from a cell. It was once cellular, maybe it's parasitic. And then it lost its cellular attributes and became this capsid-like thing. So there's all kinds of interesting ideas thinking about the origin of viruses. And your book has a blurb, maybe a page on that. I think I've asked you to read.
on, that, what they're thinking in terms of the evolution. You know, how did the viruses evolve? So kind of, you know, to generate this kind of data, you have to work with viruses, but it's challenging because you have to find the right host so you can get copies of the virus and figure out, well, how does it replicate? Especially if you want to examine the DNA, you have to get enough of it, you know, and so that, I'm not going to get into a ton of this, but when you're in the lab, it's tricky. You have to find the right cellular host, whether it's bacteria. So we've talked a lot about bacteriophages when we were looking at horizontal gene transfer ideas. Working with bacteriophages are the easiest, I would say. I've worked with cultivating bacteriophages and animal viruses, and the bacteria system is much easier. You can do it with animals too. So you work in like tissue culture, you have special plates and broth media. It's almost looks like a fancy, almost like a roux bottle. But basically you try to get these containers and the right broth media to support the host cell so that you can keep the host cell alive artificially like this. And then you infect it with the virus or the bacteriophage if it's bacteria. And you can grow it essentially by allowing it to infect the host cells. And so it's much easier to work with bacterial systems compared to animal. And plants are my least favorite. I've only tried that once, and plants are challenging to have their cells available to use for growth of the virus because you have to strip the cell wall off in order to get it to work in a lab setting. You know, it happens out in nature just fine, but we don't know we don't know a lot. I'm not that I'm a virologist. I've only done this a little bit. I'm not really well experienced with this, but a lot of it, they just don't know how to like get them to work in an artificial setting in a lab like that. It can be tricky, but they have some systems that are pretty down pat and they've been troubleshooted, and they don't have like a growth curve similar to what we already talked about with bacteria. When you grow bacteria in a batch culture and a liquid culture, and we had that growth curve. This is kind of similar, but not exactly. And they name it different things. We're not going to get into the different, you know, I'm not going to get into detail on this with viruses, but you can see the terminology is a little different. They call this, instead of a lag phase, they say it's the eclipse period, not exponential, now it's rise. And then here they're showing this is when they usually bust out of the cells. But you can get data from this. So it's similar to a growth curve in bacteria. You can still get useful data from it. It's not, you know, it's a little different, but you can see here what they were interested in is looking at, you know, well, how many phages are coming out of each infected cell, because epidemiologists need to know that so that they can start predicting spread of the virus and look at the epidemiology of it. So it's very useful information. And so they can do this with most of the viruses that infect either animals or bacteria. They can track them like this, similar to what you do with tracking how bacteria to grow. But it's tricky because you always have to have the host cell there as you do this. All right, we got to stop. We'll get more into viruses on Friday.